So let me. Our lecture for today is on biomolecules. Um, Very important. We are looking at the components that make up our body that are present in the organisms that surround us and that are present in the food we consume, right? What goes into that? What are their properties? What do they do? What is their functionality? Okay. When we're talking about biomolecules, we're usually talking about organic molecules. Has anybody here taken organic chemistry? Um, or have any of you like like heard of what an organic compound is? Nobody? Nobody? Okay. Well, organic compound, right, has to do with compounds which are made out of carbon, right? Carbon is the ideal uh, atom for life because it's so flexible. It makes four bonds, right? You could form four bonds with carbon. These four bonds can expand to structures which are gigantic in size, and they could take a variety of different forms. And then you could attach different functional groups in there to change its property. So if you wanted to use one atom to build all the structures you need for life. Carbon is the ideal substance for that. There are discussions about alternate chemistries and alternate atoms you could use to build life around. But for the conditions we are in now, there's nothing that really beats carbon that we found so far. So even if we go off in a spaceship and we look at other planets that are like us, chances are like the aliens we'll meet will be carbon-based life forms. Right? When it comes to the molecules we use carbon to build, you could divide them into a number of different categories. These include the carbohydrates, lipids, proteins, and nucleic acids. You should have heard about these in the context of nutrition, or if you were watching a sci-fi movie. So they're really common, really well-known, really broad categories. Can anybody give me an example of each of these different categories? For example, the carbohydrates. What would be an example of that?
Would glucose be an example?
Okay, good, good, good. Glucose would be an example of a carbohydrate. A lipid. What would be an example of a lipid?
Butter.
Butter, right? Butter has lots of fats in it. Fat's a type of lipid. What about protein? Anybody give me an example of a protein? Hemoglobin, right? Hemoglobin, yeah, you put hemoglobin together. Hemoglobin is composed of protein. What about nucleic acid? RNA and DNA. Okay, good, good. So yeah, you run across each of these biomolecules in your everyday life. Bread itself isn't a carbohydrate, but it has a lot of carbohydrates in it. Butter, of course, stands for lipids. Then you have protein from your steak, and then you have the helix molecule, which forms DNA. Everybody's seen that, right? So it impacts you on an everyday life. It's present everywhere. So if we dive deeper into this category, the first category that we find out we'll look into is carbohydrates. They're very simple. They consist of carbon, like all biomolecules do, at least the important ones, hydrogen, and oxygen. And you can see these structures here composed entirely of these different atoms. They're used for many different purposes. Some of them, the primary use that you're most familiar with is energy storage. So glucose, sugars, carbohydrates alike, they're the primary form of energy for at least certain parts of your body. Your metabolism centers around breaking down these molecules and extracting energy from them. But there's other purposes you could put carbohydrates through, such as structure. Some of the structural components of the body in you and other organisms can be made out of carbohydrates. So a little bit of flexibility in this molecule. So getting more specific, when it comes to chemicals, can you name a type of chemical that would be a carbohydrate? Diving deeper than just naming a food item. Let me pull up the question over here. So what would be an example of a carbohydrate out of all the options that I just put up? Okay. Good. Yeah, people got it right. Glucose would be an example of a carbohydrate. Many different other examples of carbohydrates. You could divide carbohydrates into different groups, right? First group would be the simple carbohydrates, and these would be known as sugars. They include the monosaccharides, and it should be listed here, but there's also the disaccharides too. So 1 and 2 unit components. Simplest form of carbohydrates, you can find them in these simple chains or these simple circular structures. You combine them together and they form even more complicated carbohydrates. Examples of them are glucose, fructose, and sucrose. Does anybody know what the common name for sucrose is? How about actually not, depending upon the type of corn syrup, not really. Uh, Ariana, what would sucrose commonly be known as? Do you want to take a guess at it? No guess? Okay. How about Bridget? What would be the common name for sugar? Okay, I give it away. Okay. What would be okay, I gave the answer right. What would be the components of sucrose? How about that
The
for a question?
component of what?
Like sucrose. Sucrose is table sugar, right? So what would be the components of sucrose? Sucrose is a disaccharide, right?
Mm-hmm.
So what do you think sucrose would be composed of?
Um,
I'll give you a hint. It's on this. It's on the same slide.
oh, glucose or
Glucose and what else?
fructose?
Fructose, right?
Yeah.
So good job. So sucrose is a disaccharide. It's composed of the monomers glucose and fructose, and this becomes relevant when you're thinking about tooth decay, right? So tooth decay, you know that sugar is bad for your teeth. So you have bacteria on your teeth that likes it when you have glucose in there, right? Because it could feed off the glucose. It likes it when you have fructose in there because it could feed off the fructose. But it especially likes it if it has both there, right? So if you have glucose and fructose, then it's really happy and it could grow really like out of control and cause tooth decay, like lots of tooth decay on your teeth. So you could blame eating sugar for all that damage that you have to get rid of when you visit the dentist after this lockdown occurs or is taken away, right? Right. So even these very simple monosaccharides, you can see they have many different complex properties and consequences that extend from these properties. Glucose, for example, swinging our focus back to that, it is one of the simplest sugars out there, but it has different forms, such as an alpha and a beta form, and a D and an L form. So can anybody tell me what I mean when I mean the alpha and the beta form of glucose? What is what is that? Okay. So what would be the difference between alpha and beta form? Alpha is used for energy storage, right? And beta can be used for structure. But what would be the difference between these two different molecules? Chris, what do you think? What do you think in terms? What do you think is the difference between the alpha and the beta glucose? What would you say is the difference between them? Like, if you had to guess. Okay. Um, yeah. So you say that alpha is branched and beta is not branched, but it's actually a little bit simpler than that. Can anybody give me another guess? How about Ethan? What do you think is the difference between beta and alpha glucose?
Glucose. Is it because they're like isomers? They have a different uh, composition or something?
Yeah, you're on the right track. They have a different conformation, right?
Yeah.
So you see the green lit up right over here, and you see this OH is lit up in green too. So the difference seems to be centered around this OH, right?
Right.
So what would be the difference? What would you guess is the difference between the OH in the
Uh,
alpha and the beta?
the OH is like pointing down on the alpha, right? While
Yep,
on the
yep.
beta, it's pointing up.
And then it's pointed another direction on the beta. Good job. Yes, yes, that's true. So alpha and beta, like this OH is pointed in different directions. And this might seem like a trivial thing, but actually it has chemical consequences. Alpha, you link it up together, you got starch, right? Starch is an energy source that you can use. Humans easily can digest it. Beta form, on the other hand, you have the OH sticking in another direction, and you link that up together, then you get cellulose. Can you digest cellulose? Nope. But you can use it as a building structure, right? So a lot of cellulose is in wood. We use wood to build our houses. Not much can digest our houses except for termites, right? So even this little tiny thing, it could have very very important consequences down the line if you're a homeowner, for example. Okay, what about D and L glucose? What's the big difference between those? If we have D and L glucose, what would be the big change between them? Any ideas? Okay. Hope, do you have an idea of what would the difference be between D and L glucose? You want to take a shot at it? Okay, so there's difference between the conformation of A, alpha, and beta, right? So what would be the difference between D and L glucose? Okay. Okay. Lupita, what do you think? Okay, so Lupita, can you be a little bit more? Can you be a little bit more specific than that? So the difference between alpha and beta is conformation, right? The difference between D and L is also conformation, right? Um, if you wanted to put it colloquially, what would you what would you say they are in terms of like putting it very simply? They're mirror images of each other, right? So they're kind of reflect. If you're looking at D and L glucose, they're reflections of each other. Like you have D glucose on one side of the mirror and L glucose on the other side of the mirror. They're just flipped, right? Okay, good, good. And you can see it over here. You have the cyclic form of glucose, and then you have the linear form of glucose. This cyclic form is much more common. You have the A and alpha and the beta form of glucose. The OH is on like different sides of the molecule, different like conformations. And you have D and L glucose. They're flipped images of each other. And this has consequences when you're talking about digestibility, right? Most organisms work with and digest D-glucose, whereas we can't digest L-glucose. It's just some weird thing that is synthesized in the laboratory. It doesn't really have much use except for maybe dietary supplements. They still taste sweet, but you add them in and they don't really give you much in the way of calories. Okay? Okay. Moving on, higher level, we have complex carbohydrates. So for complex carbohydrates, we have monosaccharides. Monosaccharides are linked together into these giant chains, and they kind of serve as more long-term energy storage, right? Sugar is more the immediate energy source, but if you want to store them a bit longer, then you link them up into these entire chains and you store them away, at least for some of the complex carbohydrates. Can somebody give me an example of a complex carbohydrate? Like, what would be a common example that you'd often find in everyday life? How about Kira? What do you think?
I'm sorry, can you say it one more time? Because it's like breaking in and out from my side.
What would be an example of a complex carbohydrate?
Is that like, I was gonna say starch, but I don't trust myself.
Starch.
Oh, it is?
Yeah, that's
Okay.
correct. That's correct. Good job. Good job. Yeah, starch would be a complex carbohydrate that you can yeah, serves as a food storage for plants. Others would include glycogen, which is kind of the starch for animals, but it's slightly different structure. Then you have cellulose, which is another complex carbohydrate, used more for structure than for energy storage. And these are combined together through what are known as glycosidic bonds, right? Carbohydrate linked to another group. You have your OHs come together. They have this water going away in what's known as a dehydration reaction, and then it becomes a more complex molecule over here. Okay? So keep this in mind. You'll be seeing this dehydration reaction over and over and over again. Moving on, we have lipids, right? Lipids are, you could define them as biomolecules that are solvent in nonpolar solvents. And so they don't really like to interact with water very much. You see the carbon chains over here for these fatty acids. very nonpolar, stick with each other, don't stick with water very much at all, not much for water to interact with. These fatty acids can be combined into what are known as triglycerides. When you think about fats in the common sense of like, don't eat too much fat or you'll be unhealthy, you'll often be thinking of these type of molecules. And you could create them by binding these together in a glycerol in a reaction very similar to the reaction you see for carbohydrates. They come together, they lose water, and then they're a bigger molecule. Okay. Can anybody think of examples of lipids? Steroids, right? Steroids. What would steroids be used for? One, what would be an example of a use for steroids? What is their, we know their name, but what is their functionality? If you can, answer through your mic so people don't have to use the chat window. I mean, it's fine if your mic's not working, but if you have a mic, use it. Okay. So steroids would be, steroids are signaling molecules, right? Um, what's the first thing you think of when you hear steroids? You think of the drugs, right? So the drugs, you take a bunch of steroids and you like get really strong and really powerful. And the reason for that is that steroid molecules, they mimic certain signaling molecules in your body. So like some signaling molecules say, "Oh yeah, let's get the body to this guy's supposed to be a dude, so let's get the body to develop extra muscles and stuff." And then you take these steroids in your body, and then your body thinks it's signaling that way, so you grow extra muscles, and then you become really big and strong. And then there's consequences down the line, but that's for another day to worry about, right? So in addition to energy storage, they're also like commonly used for signaling. They're commonly used for structure, right? Phospholipids are a um, very prominent component of cell membranes. Cholesterol is another steroid, another component lipid that makes up the structure of the cell membrane. So a lot of different things that lipids can do, right? So we know that lipids are very dense energy sources. Uh, we know that. What would be the reasons for this? Why are they so good at storing energy? The, just throw this question up for you guys. What makes lipids so good at packing so much energy together? Okay, I'll give a few more seconds for you guys to get your answers in. Okay. Okay. Yep. Everybody, people are on the right track. Yeah. There's two major reasons I could think of. for lipids being such a good energy source. The first is that they're non-polar, right? They could pack together very well. They pack together very well. They exclude water. On the other hand, you have these carbohydrates here. They're interacting with water all the time. So more water equals more weight. Another reason is that you look at the molecules over here, very, very reduced, don't have much oxygen associated with them, not really oxidized, where you see here oxygen's everywhere pulling away electrons. The more reduced the molecule is, the more electrons you could pass on to oxygen, the more energy you can release. That's why lipids are such dense energy sources, very good for energy storage, among other things. Okay. Next thing we're going to go over is protein. Protein, probably the most flexible category that we're going to go over. It consists of different components. The base component is called the amino acid, and you could recognize amino acid and be sure to pay attention to this. You could recognize it by its different groups attached to it. So all amino acids have this group, right? And what is this group called? What is this group over here? What would be the name of it? The amino group, right? And what would be the name of the group over here? The carboxyl group, right? And then it has a carbon at the middle, and the carbon is attached to an R group. Okay, so you have your amino group, your carboxyl group, and your R group. Correct? These together form up the amino acid. The R group gives it its property, and you could link these amino acids together in a chain. And you link it together by taking the carboxyl group, the amino group, combining them, losing a water just like you saw for the lipids, just like you saw for the carbohydrates, and forming this bond, which is known as a peptide bond, right? So you have these amino acids linked together to these chains. Can anybody tell me what the functionality of a protein would be? What is its purpose? Marlene, what do you think would be one of the purposes for protein? What could it do? Do you have any ideas, Marlene?
I was gonna say their shape determines their function, their biological function.
Can you give me like one function?
Um,
Just like shoot in the dark.
like movement, signaling, or
Yep. So
okay.
pretty much practically anything you could think of, a protein can handle it, right? They're the machines of the body. You want to you want to mediate a chemical reaction, you want to break something up, you want to build something up. Whatever the task, proteins can handle it. And it's because they have such a flexible structure, right? You have as we said before, the amino acids, they are the building blocks of the protein, and there's several different types of it. For example, you have the non-polar amino acids, right? And when you're talking about non-polar amino acids, they have these groups over here. The R groups are strands of CHs, and if you like remember what we talked about for CHs, right? They're not really interactive, so they don't really have a charge on them. You're talking about the acidic amino acids. They have acidic groups on the R groups. You're talking about basic amino acids. You have basic groups for your R groups. And then your polar amino acids, they'd have what you'd expect, polar groups, oxygens pulling electrons away and making things have charges on them, right? Making making it so that the water can easily interact with it, and like one portion of the molecule is pulling away oxygen, pulling away electrons from the other portion. So all these amino acids, they can combine together to give you a complicated structure, which I'll go over quickly over here. You have your primary structure encoded by the sequence of the amino acids. Different sequences will give you different properties. Because of this primary structure and the surrounding environment, the protein will fold up in localized areas known as the secondary structure, such as beta sheets or alpha helices. The overall folding of the structure of the protein is known as a tertiary structure. And then these polypeptide chains come together into these giant quaternary structures. And these form complicated, basic, simple at its most basic level, but really complicated structures that are machines that could perform anything, practically anything that you could think of when it comes to the needs of the body. So you have this language here that encodes these very complicated devices. And you could put these devices to work on practically anything you can imagine. Okay. And some things you can't imagine. So that's protein structure. Moving on to last but not least, nucleic acids. They're special molecules, right? And they consist of different parts, right? You have your phosphate over here, you have your sugar backbone over here, and you have your nitrogenous base, which gives the nucleic acid its identity. It's held together multiple ways. You have your, this is the DNA, right? This is a DNA molecule. You have your strand over here held by phosphodiester bonds. So you have the phosphate here and you have the ester here, phosphodiester. Yeah, but DNA is double-stranded, right? So if you have double strands, then the strands are held by the bases, and the bases form hydrogen bonds with each other. So different bonds hold different parts of the this nucleotide structure together, and the nucleic acids have, particularly DNA has a special function. It's the molecule which carries your genes, right? Your biological legacy. So it's what's responsible for passing it down. It also has some limited enzymatic activity, but not as much as proteins. So that's a secondary function of it. But for the primary reason nucleic acids, in particular DNA, exist, their storage of hereditary material. Why do you think it's so good at the job? So I'll throw that to the class. Why do you think DNA is used as a hereditary material and other molecules aren't? What makes it so great for that? Okay, so Seth, do you want to give me like what answer you think is correct? Microphone if you have it, your chat window if you don't have a microphone. Okay, so give it on the chat window then. What answer do you think is correct? Which one of the options do you think is correct or not? Do you want to take a shot? Okay, because it doesn't. Okay. Why do you think it? Because it doesn't alter pH. Why do you think that's the reason why it's such a good hereditary material? Okay. Well, um, there's actually, yeah. So it's actually, that's not actually the answer. Does anybody know another option? Anybody want to explain another
Is
option?
it because it's double stranded, so it's more stable?
Okay. Good. Yes, that would be one of the answers.
It's due to the hydroxyl mixing hydroxyl group, so it makes it less reactive in the
Okay.
nucleotide, right?
Yep. So one of the reasons DNA is such a good material is that it's very stable, right? It's actually the absence of hydroxyls that make it very stable. So you don't have like reactive oxygens going everywhere, right? And like reacting with everything. Um, how many of you have seen Jurassic Park? Or am I like showing my age here? Nobody's seen Jurassic Park? Or only a few of you? Huh. Okay. Well, does everybody know the concept of Jurassic Park? Right? Yes, no. Okay. So Jurassic Park, right? They for those who haven't seen it, they extract DNA from a mosquito, and supposedly that allows them to clone dinosaurs. This is greatly exaggerating the stability of DNA, but there is like sort of a kernel of truth to it. For certain organisms that are already dead in certain situations, you can extract some DNA and like kind of get an idea of what's going on, because DNA is compared to other biomolecules, at least it's a bit stable, and so you could leave it out in the desk a little while. It will decay, but not as completely as other molecules do. And that's one really good thing when you're thinking about hereditary molecule to could transmit your genes, right? Because you want to ensure that it will not decay, it will remain the same, so the information is accurate. Another thing that you want that contributes to its suitability for hereditary material is the fact that it's double stranded. This is another central property, right? Because above all else, when you have your genes, you want to make sure that they're transmitted accurately. And so when the DNA replicates, it has two strands. The strands could break apart, and then you have a template for synthesizing the new strand right with you, and then you use the template to synthesize a new strand, and that's your new DNA molecule. And that way, error transmission is kept at a minimal level, because if you have offspring, if you have children, right, you don't want your kid to get a mutation here and there, and all of a sudden you have a leg sprouting out of its forehead. That would be pretty bad. So you want accuracy, and accuracy is really it's really complemented by the double-stranded nature of DNA and its stability. Okay. Okay. Does anybody have any questions on what I just went over? You've absorbed it. Have you absorbed it at least partially? Does it make sense? Any questions or corrections? Okay. Good. Um, so if not, you could just go over the material again, and hopefully you'll get it more. But in the meantime, let's look at the worksheet we have over here. And so this is worksheet 3, right? And we just went over like basically all the material you would need to understand this worksheet. What you have to do in this worksheet is take these different molecules, right, and use it to construct the molecules that they're talking about over here, right? So the very first thing that we want to discuss is what you would use to make a carbohydrate, right? Specifically, in this case, starch. Do we want to use to make starch? So, what would you guys pick? Any volunteers? Out of all these different numbered chemicals, what would you pick to make starch? Emily, do you have any ideas?
No, I'm not sure.
Okay. Starch. Well, starch. Let's take it step by step, right? Starch is a what? What type of chemical? What type of biomolecule is it?
Um,
So you have your carbohydrates, your proteins, your nucleic acids, right? Which one of those would be starch?
sorry, repeat that again.
So you have carbohydrates, proteins, lipids, and nucleic acids. Which one is starch?
Carbohydrate.
Okay. And carbohydrates are made out of are complex and simple, right? What are simple carbohydrates?
Um,
Sugars, right? So
yeah,
sugars are simple carbohydrates. Does this look like a sugar to you? Carbohydrates are carbons, hydrogens, and oxygens, right?
yeah. I mean, it
So
could
does,
be.
does this look like a sugar to you?
I don't know because there's a lot of them that have carbon, hydrogen, and oxygen in them.
Okay. Yes, that's true. But is starch is a complex carbohydrate, correct?
Yeah,
Okay. So if it's a complex carbohydrate, it's a chain of carbohydrates, right?
yeah.
So since it's a chain of carbohydrates, would it be made of the same molecule or a different molecule? Based upon what you see here. Are these the
Um,
same molecules or
yeah,
different
the
molecules?
same.
Same. Okay. So since it's the same molecule, we want to look at something that appears twice on this
Mhm.
worksheet, right? So which one of these appear? Which one out of all these chemicals? Which one appears twice?
Oh, 7 and 11.
Okay. Okay. 7 and 11. Looking at it, are these exactly the same?
No.
Nope. Okay. Then what would be the same?
Um,
Let's look at 11, right? Do you see 11 anywhere else on this worksheet? And
um,
keep in mind, sometimes molecules are represented slightly differently. Which one would be identical to 11 here? How about this one, right? Does this look identical to you?
I mean, not really, but I guess.
Okay. Keep in mind, right? In some, and this is important to keep in mind, some representations of molecules are different. Like here, they don't explicitly list all
Oh,
the
yeah, they
hydrogens.
don't have the hydrogens.
Yeah, okay. So 11 and 13 together, right, would be
Mhm.
able to form the carbohydrate starch. Good job. Good job. Okay, good, good. Okay. So you have starch from 11 and 13. Okay, lipids, triglycerides. What will we use to form the triglyceride? Any any suggestions for me? What could we use to form a triglyceride?
Maybe one or two, maybe.
One or two, right? And that would be the fatty acid of the triglyceride, right? What else could we use to form the triglyceride? Triglyceride, right? So what is this part? What would this part be? Let's look at our lipids, right? Let's look back at our lipids. We have our fatty acids, right? And then what other portions are there? Nine. Okay, 9. Good, good. So what would 9 be called? If on your microphone if you have it, chat if you don't, but I prefer microphone.
A glycerol?
Glycerol, right? Good. So glycerol, combining these fatty acids, triglyceride. Okay? Yeah. And you could mix and match them if you want. You could have 3 of this, whatever. What is the difference between 1 and 2? Can anybody tell me? Think back when you're
Is it the double bond on one between two carbons?
okay. And what is that in terms of the nutrition label? What would that mean?
Okay. I don't know.
You see, okay, lipids are fats, right?
Right?
Okay. What sort of fats are you looking for in the nutrition label?
Saturated?
Saturated and what
Oh.
else?
Saturated.
There's saturated and
Non-saturated?
unsaturated fat, right?
Okay.
Well, there's different types of unsaturated fat, but we won't get into that. Okay, which one do you think is saturated?
One.
Okay. How is it saturated?
And I don't know.
When we mean saturation, right? It means it has tons of something, right? Let's say tons of hydrogen. Which one is saturated with as much hydrogen as it can take? 1 or 2?
Oh, two.
2, right?
Yeah.
2 is a saturated fat, right? So you don't want to, at least according to some people, you don't want to eat a lot of saturated fats, no matter how fun it is, right? So this would be a saturated fat. This would actually, well, not in and of itself, but this would be one of the components of saturated fats. This would be an unsaturated fat, right? You combine to get them together with a glycerol, then you could form your triglyceride. Okay, good. So that would be for lipids. And then what would be the bond that you form between these fatty acids and the glycerol? What would that be called? What would be the name for it? Any ideas? Okay. It would be an ester bond, okay? Okay. Now moving on to protein, right? Protein, you want protein with, you know that proteins are, yes, esters. You know proteins are composed of different amino acids. And so you need to use these particular amino acids to construct your protein. A nonpolar amino acid, a hydrophilic amino acid, and an acidic amino acid. Okay? So first off, we need to, in order to filter this out a little bit and like break down our filter as to what molecules may or may not be amino acids, we need to determine what an amino acid looks like, right? What do we look for when we look for an amino acid? What components are we looking for? Okay, I try not to pick people twice, but I can't remember who I picked. So if I pick you twice, I apologize. Nitrogen, right? Nitrogen would be part of what? What group? Remember, microphone if you have it, if possible. Okay. Amino group, right? And then, okay, what else would you look for?
Would you be looking for a carboxyl group as
Okay.
well?
Okay. And then a central carbon, right? So does this fit the bill? Number 3? Okay. Amino group, right? Central carbon, and then your carboxyl group. And then you have a R group over here, correct? So this would be an amino acid. What about 4? Yes or no? Amino acid or not amino acid? Nope. What about 5? Amino acid or not amino acid? No. What about 6? Yes or no for amino acid? Amino group, carboxyl group, and then your R group, right? So amino acid. What about 7? Yes or no? Someone other than Juan for once. No offense, Juan. Okay. No, no. Okay, how about 8? Yes or no, amino acid for 8?
Yes.
Okay, yeah. Amino group, carboxyl group, R group. What about 9? 10? No. 11? 12? No, no. What about 13? 14? Yes or no for 14? You have your carboxyl group, right? And your amino group and your R group. Okay, what about 15? Okay. So 14, 6, yeah, okay. So yeah. So we have our amino acids and non-amino acids, okay? And okay, which one of these amino acids would be our non-polar amino acid? Bridget, which one do you think out of all the amino acids that we listed, which one would be the non-polar amino acid?
Which one would be the what?
The non-polar amino acid.
6.
A 6, right? Why do you think it would be non-polar?
I'm not quite sure.
Because of the R group, right? The R group is CH3.
Oh, I see.
Okay, so that would be non-polar. What about the hydrophilic group? What would be a hydrophilic amino acid?
5. Wait, no.
So we learned to recognize our amino acids, right? Which one of them would be polar? Okay, you see the amino group right here in the carboxyl group, right?
Yes.
This is the R group, right? Does this look polar or non-polar to you?
Polar?
Polar, right? Because of the oxygen
Mm-hmm.
pulling away. So this is a polar group, and so since it's polar, it'd be hydrophilic. What about the acidic group?
14.
Where would the R group be located in this molecule?
Wait, which one?
The what? So you have your amino group and your carboxyl group here. So this would be the functional group, right?
Yeah.
Okay, so does this look acidic to you based upon what we read and we talked about before?
Yes.
Yes, right? So this would be an acidic group. So nonpolar, polar, acidic amino acid, okay?
Okay.
So you have all your amino acids over there. Okay, good, good. Can anybody tell me what bond these amino acids would have together? Peptide bond. Okay, good, good, good. Okay, next one, nucleic acid. Let's have a question for that. Fourteen would be a basic amino acid, I think. I think it's basic. Yeah. You can check up, but I think it's basic. So pick out the correct sugar to form that you use to form DNA. Out of all these molecules, what would you use as a proper sugar to form DNA?
Oh, it's a 10.
Okay, right. Okay, a couple more seconds for people to.
Oh, sorry.
Okay, yeah, good. Um, 10 would be the base, would be the sugar backbone to form your DNA. What else would you need? You have your sugar backbone, right? What else would you need to form this guanine that they're talking about? The what?
Is it number 12, the nitrogenous base?
Okay, so you have a nitrogenous base attached
Yeah.
to your sugar backbone. What else?
Yeah,
Do you
and
have?
then you'd need to have a phosphate group as well. So I'm thinking 5.
Okay, right. So phosphate, backbone, and base form together to form your guanine, right? And what would be the bonds that hold them together?
Is it phosphodiester? Or, no, no, no.
Phosphodiester would bind together the backbone, right? And then what would bind together the different strands of the DNA?
Is it a hydrogen
Okay,
between
good,
the
good,
base pairs?
good, good. Yeah. So, I mean, for the strand itself, the backbone, phosphodiester bonds, and if you, yeah, and if you want to see what it looks like, then You could look, see over here. And then the bases are held together by hydrogen bonds. Okay? So, okay, I recommend for the sake of your ability to absorb the material, I just went over this like in a descriptive sense to save time. But if you really want to practice, you should draw out the structures and how they bind together, right? That way it will really sink into you. Also, when you're doing the worksheets, make sure that you do them thoroughly, right? I see some people might be missing like a component of a question here or there. When I go over these, I think last time I forgot to circle the polar component of one compound, but even if I miss it, try to get that done. Okay. So draw if you can just draw out the structures if for nothing else to be able to absorb the material more readily. And I guess that would be it for this particular worksheet. Does anybody have any questions on it? Any clarifications that you need to talk about or corrections? If any mistake I know that you noticed, everybody's all good. Okay, so if nobody has any more questions, then that's pretty much it for this discussion. I'll stick around for a little while. You could throw me any questions you want. You could you could ask me in email or you could ask me here. I've already uploaded this assignment, so be sure to turn it in through iLearn by the due date. And I'll be making an announcement about it shortly. Okay, so I guess that's it for today. If you have any questions, ask me. If not, have a great day. Okay. Okay, so if you want to know how the peptide bonds link the amino acids together, let's just go back to our slide, right? You have the amino acid here. You have the amino group. Amino group goes into the carboxyl group. So it's kind of like Legos, right? You just like plug into here, plug into here, plug into here, plug into here, and then you have a strand that goes off for as long as you want. And then these are the peptide bonds between each of the different amino acids. Okay. Any further questions? So yeah, this is kind of an unstructured class. Technically, you don't have to do as much as you would normally have to do, but to get the most out of it, you might want to like do stuff like draw out the different structures and see how they link together. I think for the quiz that he just posted, the average score was quite a bit lower than we were expecting. So yeah, you might want to keep that in mind as you figure out how you're going to practice for the next quiz that's coming up. Make sure that you absorb the material properly. Oops. Uh,